Okay, let's unpack this. Have you ever found yourself wondering about those viruses that seem to have mastered the art of living with us for life? They're incredibly common, persistently tricky, and they affect millions worldwide. Today, we're taking a truly deep dive into a topic that fits that description perfectly. Herpes simplex virus infections.
Right.
We're talking HSV1 and HSV2. This isn't just a surface level chat. We're going to explore every single facet of HSV, from its intricate biology and how it interacts with our immune system, to its pervasive global impact and the cutting edge of current therapies.
Yeah.
Our mission is to arm you with the kind of comprehensive, detailed understanding that elite professionals in this field truly grasp.
And what's truly fascinating here is how these viruses, despite being among the most well-studied pathogens, they continue to present such complex challenges, in terms of both treatment and prevention.
Exactly.
Our source materials, specifically Chapter 192, it's an incredible foundation for this journey. It really lets us peel back the layers and understand HSV on a cellular and systemic level.
That's right. So if you're ready to go beyond the basics and truly master the nuances of HSV, from its viral genome and replication cycle to clinical management and that incredibly complex dance between the virus and our immune system, then buckle up.
Yeah.
This is going to be a deep dive like no other. Unveiling the master of persistence. Herpes simplex virus fundamentals. So let's begin right at the fundamental level. What exactly are we talking about when we say herpes simplex viruses, or HSV? We often hear about HSV1 and HSV2, historically known as herpes virus hominis. What makes these particular viruses so noteworthy?
Well, these aren't just one-trick ponies, causing a single type of ailment. HSV1 and HSV2 are, in essence, highly versatile agents. They're capable of producing a really wide array of infections throughout the human body.
Okay.
Our source material really underscores this versatility. They primarily affect what we call mucocutaneous surfaces.
So skin and mucous membranes, like mouth, genitals.
Exactly. Think of your skin and all those mucous membranes.
Yeah.
But their reach extends far beyond that. They can infect the peripheral nervous
The
system.
nerves outside the brain and spine.
Right, those nerves. And critically, they can also invade the central nervous system itself, so the brain and spinal cord. And, occasionally they can even target internal visceral organs.
Wow.
A crucial takeaway from a professional standpoint, and the source material is clear on this, is that because of this wide range of potential targets, and frankly the severity of some of these infections, prompt recognition and rapid effective treatment aren't just advisable.
They're essential.
They are absolutely vital. Why? Because they directly reduce the morbidity that's the disease and its complications, and also the mortality rates associated with these infections. This is about preventing long-term health issues and, in severe cases, saving lives.
That puts it into perspective immediately. These can be very serious. And to truly understand how such a virus operates, we need to examine its very blueprint. So let's get into the etiologic agent itself. This is where it gets really interesting because we're talking about a remarkably sophisticated piece of biological machinery, aren't we?
Absolutely. Think of the HSV genome as like an incredibly detailed instruction manual, or maybe a complex software program. It's a large 152 kilobase linear double-stranded DNA molecule.
152 kilobase, that's pretty big for a virus.
It's quite substantial, yes. And it's packed with information. This blueprint encodes over 90 different transcription units.
Which are copied into RNA.
Right, segments of DNA copied into RNA. And from those, at least 84 identified proteins are made. Imagine that. 84 distinct proteins, each with a specific function, all orchestrated by this single DNA molecule.
84 proteins. That's a lot of moving parts for a virus. And I know there are two main types, HSV1 and HSV2. How similar or different are their blueprints?
That's a great question, and it speaks to their shared lineage. While their overall genomic sequence homology, meaning the exact DNA sequence similarity, is around 50%, the proteins they produce, what we call their proteome, are remarkably more similar.
Oh, interesting.
Yeah, over 80% homology in the proteome. So they're like siblings, where their genetic code might look a bit different on paper, but the actual proteins they build are largely antigenically related.
Okay.
This means our immune system often recognizes significant portions of them as similar.
So, like cousins who share many facial features, even if their internal genetics aren't entirely identical?
That's a very apt analogy, yes. And within that similarity, there are critical differences. There are type-specific regions unique to HSV1 and HSV2 proteins.
Ah, the unique identifiers.
Precisely. These unique identifiers are incredibly important for how our immune system responds. They're what allow our bodies to distinguish between the two types, and critically, they've been harnessed to develop advanced serologic assays.
The blood tests.
Exactly, the blood tests that can reliably tell us whether someone has antibodies to HSV1 or HSV2. This is crucial for accurate diagnosis and understanding past exposure. And beyond simply distinguishing types, for epidemiology, tracking how the virus spreads, researchers use techniques like restriction endonuclease analysis, or even direct sequencing of the viral DNA.
Getting really granular.
Very granular. This allows them to differentiate not just between HSV1 and HSV2, but also among different strains within a single subtype. This level of detail is incredibly powerful. It allows us to infer epidemiologically related sources.
Like tracking transmission between partners. Or in an outbreak.
Exactly. Helping us track transmission between sexual partners, mother-infant pairs, or pinpoint common source outbreaks. And here's a layer of complexity that really underscores the virus's adaptability. Recombinant viruses.
A mix of HSV1 and 2.
Sort of a genetic mix and match, yes. They actually circulate in nature. Furthermore, deep sequencing studies have unveiled that a single individual can harbor more than one variant of HSV1 or HSV2 simultaneously.
Wow.
It truly paints a picture of a dynamic and diverse viral population living within us.
That's truly mind-blowing. So it's not just one virus, one person. It's potentially a whole ecosystem of viral strains even within a single individual. That incredible blueprint, all those proteins, how is this sophisticated package actually put together? What does the viral particle itself look like?
Right. Once the viral DNA is replicated, it's very precisely packaged within a regular icosahedral protein shell. This is called the capsid.
Like a geometric shape.
Exactly, an incredibly symmetrical structure made up of 162 individual protein subunits called capsomeres. Think of it as a beautifully engineered microscopic geodesic dome. Surrounding this capsid is the outer covering, a lipid-containing membrane known as the envelope.
And it gets that from us.
It does. This isn't something the virus builds from scratch. It's actually acquired as the DNA-containing capsid buds through the internuclear membrane of the host cell. It essentially steals a piece of our cell's own machinery for its outer layer. And then, in between that inner capsid in the outer lipid envelope, there's a crucial layer called the tegument. This tegument contains various viral proteins that are absolutely essential for the very early stages of infection. It's almost like a prepackaged toolkit the virus brings along.
So it's like a finely tuned, multi-layered stealth bomber designed for invasion. How does this highly coordinated invasion, this replication cycle, actually play out once it successfully enters one of our cells?
It's a masterclass in biological precision, really, with distinct nuclear and cytoplasmic phases. It all kicks off with initial attachment and entry into a host cell.
How does it grab on?
This is orchestrated by two key viral proteins on the virus's surface, with sugar molecules attached, glycoprotein C and glycoprotein B. They initiate the process by interacting with specific cellular receptors on our host cell surface, particularly those that resemble heparin sulfate.
Like docking clamps.
Kind of like initial docking clamps, yes. But this forced interaction isn't enough. A second, even more critical step involves another viral glycoprotein D, which binds to specific cellular co-receptors.
More specific targets.
Yes. These co-receptors belong to families like the tumor necrosis factor receptor family or the immunoglobulin superfamily, specifically the nectin family. And the crucial insight here, the source material emphasizes this, is the ubiquity of these receptors.
They're everywhere.
They are found on almost all human cells. This universal key lock mechanism is precisely why herpes viruses have such an incredibly wide host range, enabling them to infect so many different kinds of cells in the human body.
And once it gets that critical foothold and slips inside, it's not subtle about taking over, is it? It immediately starts to hijack the cell's own machinery.
Exactly. Once the viral envelope fuses with the host cell membrane, the nucleocapsid, that DNA-containing core, enters the cytoplasm. From there, several viral proteins, the ones that were in that tegument layer we mentioned,
The toolkit.
right, the toolkit proteins, are immediately released from the virion. Some of these proteins are astonishingly efficient at immediately shutting off the host cell's own protein synthesis.
How do they do that?
Well, for example, by triggering the degradation of cellular RNA. It essentially turns off the host's ability to make its own vital components, effectively diverting all the cell's resources to the virus.
Wow.
Simultaneously, other viral proteins get to work activating the transcription of what we call the immediate early genes of HSV replication. These are also known as OFA genes.
Alpha genes, okay.
These OFA gene products are absolutely crucial because they are necessary for the synthesis of the next group of polypeptides, the OFA polypeptides. Many of these OVG proteins are regulatory proteins, and critically, they are enzymes essential for viral DNA replication.
Like the ones our drugs target.
Precisely, such as viral thymidine kinase, or TK, and viral DNA polymerase. And here's a vital point for us in the medical field. These enzymes, TK and DNA polymerase, are precisely the targets for most of our current antiviral drugs. Then there's a third class, the cogenes, or gamma genes. These encode the viral structural integument proteins. Their expression generally requires that viral DNA replication has already occurred. It's a highly organized sequential takeover.
It sounds like a remarkably organized assembly line designed to maximize efficiency. Once all these viral components are manufactured using our cells' resources, how does the virus put itself back together and then get out to infect other cells?
Right, so once the new viral DNA has been replicated and all the necessary proteins have been synthesized, the nucleocapsids, those shells containing the newly replicated viral DNA, are assembled inside the host cell's nucleus.
Inside the nucleus?
Yes. The process of envelopment then occurs as these newly formed nucleocapsids bud through the inner nuclear membrane, acquiring that lipid envelope we discussed earlier into the paranuclear space.
So it wraps itself on the way out of the nucleus?
Essentially, yes. Once they're enveloped, these complete infectious virions are efficiently transported through the cell. This journey takes them via the endoplasmic reticulum and the Golgi apparatus all the way to the cell surface. And from there, they can be released to infect new, unsuspecting cells.
A constant cycle.
A constant cycle. And interestingly, during this intense viral replication, especially within the nucleus, two distinct types of inclusion bodies can form in the host cell's nucleus. You might see these under a microscope.
What are they?
There are type A basophilic Fulgin positive bodies, which are actually sites containing viral DNA, signaling active replication. Then there are eosinophilic inclusion bodies, which are devoid of viral nucleic acid or protein. These are more like a cellular scar or remnant, representing changes or debris caused by the viral infection.
Yeah.
Now, perhaps the most defining and frankly challenging characteristic of herpes simplex viruses is their ability to establish latency.
Right, the hiding phase.
Exactly. This is a repressed state where the viral genomes are maintained, often for the lifetime of the host, by specific neuronal cells. In this latent state, only a very limited number of virus-encoded RNAs are transcribed. And critically, infectious virus cannot be isolated using standard methods.
So you can't just culture it out?
Nope. This means you can have viral DNA and RNA present in nerve tissue, but the person isn't actively shedding infectious virus or showing symptoms. It's like the virus has gone into deep hiding.
So the virus is there, it's present in the body, but it's just chilling out, not actively replicating or causing disease.
That's the essence of it, yes. But it's not a permanent sleep. From this latent state, a process we call activation or reactivation can occur.
The flare-up.
The flare-up, exactly. This leads to the normal, highly regulated viral gene expression, full viral replication, and ultimately the release of infectious HSV particles. This release involves a complex process called anterograde transport, where the virions travel down the length of the neuronal axons all the way back to the mucocutaneous surface.
Back to the skin or mouth.
Right. In experimental animals, we've observed that various factors can trigger this reactivation. Exposure to ultraviolet light, periods of systemic or local immunosuppression.
Like getting sick or stress.
Exactly. Illness or stress, and even physical trauma to the skin or the ganglia where the virus is residing. A critical component in understanding this complex dance between latency and reactivation are what we call latency-associated transcripts, or LATs.
LATs.
Initially described as three non-coding regions, we now understand them as a more diverse set of non-coding RNAs and microRNAs, or miRNAs. These LATs are specifically found in the nuclei of latently infected neurons.
And they're important for staying latent.
Well, studies where portions of the LAT region were deleted showed a reduced efficiency in later reactivation of the virus. And here's a truly fascinating insight from the source material. If you swap HSV1 LATs for HSV2 LATs, it actually induces an HSV1 reactivation pattern.
Whoa.
Yeah. This powerfully suggests that the LA region of the genome primarily maintains rather than establishes latency. It keeps it quiet, but doesn't necessarily set it up. Furthermore, viral miRNAs, which are part of the LATs, appear to play a crucial role in silencing the expression of key neurovirulence factors like infected cell protein 34.5, ICP 34.5. They also bind in an antisense way to the messenger RNA of immediate early protein ICP0, preventing its expression, which is vital for HSV reactivation.
So they're actively suppressing the wake-up signals.
That seems to be a key part of it. However, despite these significant discoveries, the full molecular mechanisms of HSV latency are not fully understood, and currently, strategies specifically aimed at permanently interrupting or maintaining latency in neurons are still very much in developmental stages. This is a frontier of ongoing research.
What's truly fascinating here is this idea of the virus being mostly suppressed, but still dynamic. It sounds like it's never truly asleep, maybe always just on the cusp of waking up somewhere.
You've hit on a critical nuance. While latency is indeed the predominant state for the virus on a proneuron basis, the remarkably high frequency of oral and genital tract reactivation for both HSV1 and HSV2 strongly suggests that the viruses are rarely, if ever, truly quiescent within the entire biomass of ganglionic tissue.
So not all nerve cells are quiet at the same time.
Exactly. It's more accurate to say the virus exists in a dynamic equilibrium. While mostly suppressed, there's a continual low-level activity where individual neurons show varying degrees of viral transcriptional activity, even if only a few of these infected neurons actually give rise to full clinical reactivation. There's also increasing recognition that HSV infection of the autonomic ganglia, which control involuntary bodily functions, plays an important role in both initial infection and subsequent reactivations.
Like gut function.
Yes, for instance. In fact, animal studies have even linked deaths from HSV2 infection to autonomic dysfunction of the bowel, highlighting the broader, often unrecognized impact beyond just the visible mucocutaneous lesions. And this dynamic, often silent nature is absolutely key to understanding viral spread. Both HSV1 and HSV2 are shed subclinically.
Meaning without symptoms.
Without any outward symptoms, correct. What this means is that most people infected with HSV2 and also HSV1 from oral sites experience frequent, brief bursts of reactivation. These bursts often last for a very short period, typically only 2 to 6 hours.
Just a few hours. That's incredibly short.
It is. And during these brief periods, the host's tissue-based immune system can often contain the viral reactivation locally within the tissue, often before any clinical symptoms or visible lesions actually develop.
So the immune system wins that little skirmish.
Often, yes. But this silent shedding, where infectious virus is present on mucosal surfaces without any outward sign, is a major, often overlooked factor in the continued transmission of HSV. The battle within. Pathogenesis and immunity in detail.
That makes perfect sense. So if you follow this thread of the virus as a master of persistence, let's delve into its journey through the body, what we call pathogenesis. How does it get in, and then where does it go to set up shop?
Right. It all begins with exposure. When HSV, whether it's HSV1 or HSV2, encounters mucosal surfaces like those in the mouth or genitals, or even abraded skin where the protective barrier is broken.
A cut or scrape.
Exactly. It gains entry into the cells of the epidermis and dermis. That's where the initial viral replication takes off. It's a crucial point that HSV infections are often acquired subclinically.
So you might not even know you got infected initially.
Precisely. The initial infection might happen without the person ever knowing or without any noticeable symptoms. But regardless of whether that acquisition is clinical or subclinical, it's always associated with enough viral replication to allow the virus to infect the very delicate sensory and/or autonomic nerve endings present in the tissue.
So it gets into the nerve endings, and then what? How does it make that long journey to establish latency in the ganglia?
Once inside the nerve cell, the virus, or more accurately its nucleocapsid, that inner core with the DNA is transported intraaxonally.
Down the nerve fiber.
Exactly. Imagine the axon as a long microscopic highway within the nerve cell. The viral particles literally tether onto cellular proteins that act like tiny motors, pulling them along the microtubules, which are the structural tracks within the axon, all the way to the nerve cell bodies located in the ganglia.
Like a little train.
Kind of like a little train, yeah. In humans, this transit interval, this rapid spread to contiguous neural tissue, happens remarkably fast. From those ganglia, the virus has a couple of key mechanisms for dissemination, for spreading out. The most commonly understood is the centrifugal migration of infectious virions via peripheral nerves.
Traveling back down the nerve highway.
Essentially, yes, back down to the surface. This mechanism helps explain why primary infections can involve a large surface area and why new lesions might appear quite distant from the initial site. It also explains why we can sometimes recover virus from neural tissue far from where the initial infection occurred. But there's another important route. Recent studies have shown HSV viremia.
Virus in the blood.
Virus in the bloodstream, yes. In about 30% to 40% of people experiencing a primary HSV2 infection. This viremia offers another mechanism for the virus to spread more widely throughout the body, potentially reaching diverse sites.
And where exactly does the virus prefer to set up its hidden home for latency? Are there specific spots it favors?
Yes, there are specific hideouts. Latent infection with both HSV subtypes has been definitively demonstrated in both sensory and autonomic ganglia. For HSV1 infection, the trigeminal ganglia.
Nerves for the face and mouth.
Correct. Those are most commonly infected. From there, it can extend to the inferior and superior cervical ganglia. In the case of genital infection, the sacral nerve root ganglia, specifically S2 to S5, are most commonly affected because these nerves innervate the genital area.
Makes sense.
But it's not just sensory nerves. Autonomic ganglia, pelvic nerves, and vaginal nerve roots are also commonly infected. This underscores the broad neural involvement and potential for impact on involuntary functions. After the primary disease resolves, it's important to understand that you can no longer culture infectious HSV from the ganglia. That's the latent state.
It's hidden.
It's hidden. However, the neuronal infection, defined by the persistent presence of viral DNA, remains deeply embedded in those ganglionic cells within the anatomic regions of the initial infection. This is what we call the post-primary latent state.
Okay.
The precise mechanism of reactivation from this deep latency is still an area of intense research, although we are increasingly identifying limited viral genes or microRNAs that are transcribed in latently infected neurons and appear to play a role. There's also compelling evidence of viral antigen and activated host T cells at the ganglia themselves and in the peripheral tissue.
So the immune system is still aware, even during latency.
It seems so. These immune responses, both in the ganglia and in the peripheral tissue, appear to profoundly influence the frequency and severity of HSV reactivation. In fact, this is where the immune system's role becomes even more fascinating. HSV-specific T cells have been recovered directly from peripheral nerve root ganglia. Many of these are resident CD8 plus T cells, often found right next to latently HSV1 infected neurons in the trigeminal ganglia.
CD8 plus T cells, the killer cells.
The killer T cells, yes. And these T cells are truly remarkable. They can actively block viral reactivation through at least two mechanisms. First, by releasing interferon gamma, or ISN, which has broad antiviral effects.
Right.
And second, by a process called granzyme B-mediated degradation of the immediate early protein ICP4, which is crucial for the virus to replicate.
So they're actively policing the latent virus.
They appear to be. What's even more intriguing is the inverse correlation observed here. There appears to be a latent viral load in the ganglia that correlates positively with the number of neurons infected and the rate of reactivation, but inversely with the number of T cells present in that same area.
So more virus, fewer T cells, or more T cells keep the virus load lower.
It seems more T cells correlate with lower reactivation despite potentially higher viral load. It raises a key open question that researchers are still trying to answer. Do reactivating stimuli transiently suppress these vigilant immune cells, or do they independently upregulate aphlitic genes, the genes that cause active viral replication, or perhaps both? It's a complex dynamic. This subtle interplay continues right at the mucosal surface, where the host tries to contain the virus. Once the reactivating virus reaches the dermal-epidermal junction of the skin, there are essentially three possible outcomes.
Okay, what are they?
First, and ideally, there can be rapid host containment of the infection very near the site of reactivation. The immune system nips it in the bud.
Good outcome.
Best outcome. Second, a small amount of virus can spread into the epidermis, leading to what's called a microulceration, which is associated with low titer subclinical shedding, meaning the virus is present and infectious, but there's no visible lesion.
The silent shedding again.
Exactly.
Yeah.
Or, third, this is what we see clinically, there could be widespread replication and necrosis of epithelial cells, leading to a full-blown clinical recurrence, characterized by the classic skin blister and subsequent painful ulceration. Histologically, if you were to look at a herpetic lesion under a microscope, it's quite distinctive. You'd typically see a thin-walled vesicle or an ulceration in the basal region of the skin.
Okay.
A hallmark finding would be multinucleated cells, which often contain those intranuclear inclusions we discussed earlier, along with clear signs of necrosis or cell death. and an acute inflammatory response.
Does it scar?
Usually not. A key feature that helps distinguish them, both clinically and histologically, is that once viral replication is restricted, the reepithelialization, the healing of the skin, occurs typically without forming a permanent scar, unlike many other skin injuries. And while DNA analysis of sequential HSV isolates, or those from multiple infected ganglia in one individual, usually shows similar patterns, indicating a primary infection with one strain.
Mostly the same virus.
Mostly. But more sensitive genomic technologies are now revealing an even greater complexity. We're increasingly seeing evidence of multiple strains of the same HSV subtype existing simultaneously within a single person.
Multiple strains at once.
Yes. For example, in severely immunosuppressed patients, we found individual neurons infected with multiple strains, some drug susceptible and some drug resistant. This suggests that ganglia can actually be reseeded during chronic infection.
Wow.
And because exposure to mucosal shedding is relatively common over a person's lifetime, current data suggests that exogenous infection with different strains of the same subtype can and does occur.
So you can get reinfected with a different strain.
It seems possible, yes. However, the precise role that this strain variation plays in the varied patterns of disease reactivation we see from person to person is still an area that's not fully understood.
So we have this sophisticated virus that can hide and then reactivate in fascinating ways. What does this all mean for our body's actual defense system? How does our body fight back against such a clever, persistent adversary, especially with this constant hide and seek?
That's where the immune system comes in, and it's a monumental battle. Host responses significantly influence every single aspect of HSV infection. From the initial acquisition of the disease and how severe the primary infection becomes, to our ability to resist the establishment of latency, the maintenance of that latency, and ultimately the frequency of recurrences.
It's involved everywhere.
Absolutely everywhere. And when we look at the body's defenses, T-cell mediated immunity is actually more critical than humoral immunity, which involves antibodies.
Really? More than antibodies. That seems counterintuitive.
It does. But individuals with deficits in humoral immunity, for example, those with agamoglobulinemia who can't make antibodies, surprisingly do not experience more severe and extensive HSV infections than those with T-cell deficiencies. In fact, experimental studies where lymphocytes were intentionally removed clearly indicate that T-cells play a major role in preventing lethal disseminated disease, even though antibodies do contribute by helping reduce viral titers in neural tissue. It's also important to note that some clinical manifestations of HSV actually appear to be related to the host's immune response itself, rather than just direct viral damage.
Like friendly fire.
Sort of like friendly fire.
Right.
A prime example is the stromal opacities associated with recurrent herpetic keratitis in the eye. This is a T-cell-driven immunopathologic process, meaning the damage is caused by the immune system trying to clear the virus, leading to inflammation and scarring in the cornea. Now, speaking of antibodies, the viral surface glycoproteins are major targets for them. Antibodies targeting these glycoproteins can mediate neutralization, directly inactivating the virus, and also antibody-dependent cell-mediated cytotoxicity, or ADCC.
Where immune cells kill infected cells tagged by antibody.
Precisely. In experimental infections, monoclonal antibodies to HSV viral glycoproteins have shown promise, conferring protection against subsequent neurologic disease or ganglionic latency. But large controlled studies in humans to assess their impact on disease reactivation are still lacking. The cellular and cytokine responses against HSV are incredibly complex and multifaceted. Multiple cell populations are involved.
Not just T cells.
Not just T cells, neutrophils, macrophages, a whole variety of T lymphocytes. The lymphokines, such as interferons, which are signaling proteins generated by T lymphocytes, also play a crucial role. We know from animal studies that the passive transfer of primed lymphocytes, meaning T cells that have already seen HSV, can confer protection from subsequent HSV challenge. For maximal protection, you typically need the activation of multiple T-cell subpopulations, including cytotoxic T cells, the killer cells, and T cells responsible for delayed hypersensitivity. These cells, in turn, can confer protection by releasing lymphokines like interferons in response to viral antigen stimulation.
So the T cells orchestrate a lot of the response.
They really do. These lymphokines then have direct antiviral effects and also activate and enhance a variety of other specific and nonspecific effector cells. But HSV isn't just a passive target, it's a cunning adversary. The HSV variant itself contains a variety of genes specifically directed at inhibiting our host immune responses.
It fights back against the immune system.
It absolutely does. One key example is the gene ICP47, which encodes a protein that can bind to a cellular transporter protein called TP1. This binding reduces the ability of HSV peptides, fragments of viral proteins, to bind to human leukocyte antigen class molecules.
The molecules that show antigens to T cells.
Exactly.
Yeah.
So why is this important? It effectively reduces the recognition of viral proteins by our cytotoxic T cells.
Yeah.
Essentially, it helps the virus hide from a crucial part of our immune system surveillance.
A cloaking device.
A biological cloaking device, yes. While this viral evasion effect can be overcome by the addition of interferon gamma, this reversal takes 24 to 48 hours.
Giving the virus a head start.
A critical window, yes, to replicate and invade other host cells before the immune system can fully respond. Furthermore, the entry of infectious HSV1 and HSV2 has been shown to directly inhibit several signaling pathways of both CD4+ and CD8+ T cells, leading to their functional impairment in killing infected cells and influencing the spectrum of their cytokine secretion. This is a very sophisticated viral counterattack. This intricate understanding of immune evasion also opens doors for promising new vaccination strategies. For instance, therapeutic vaccination with a replication defective HSV, specifically one that lacks glycoprotein DGD.
A key surface protein.
A major neutralizing protein target, yes. This approach has been shown to produce enhanced ADCC activity and a subsequent reduction in reactivation in guinea pig models of HSV2. This suggests that immune responses targeting cell-associated virus, rather than just free virus particles, may play a very important role in achieving full disease resolution.
So it's not just about stopping the virus from getting in, but also profoundly about how the body deals with it once it's inside our cells, actively fighting these clever evasion tactics.
Precisely. And in that context, HSV-specific CD8+ T cell responses appear to be an absolutely critical component in clearing the virus from lesions. We see this clinically.
Yeah.
Immunosuppressed patients who experience frequent and prolonged HSV lesions often have fewer functional CD8+ T cells specifically directed at HSV.
Okay.
What's even more remarkable and a true insight from recent research is that HSV-specific CD8+ T cells have been shown to persist in the genital skin at the dermal-epidermal junction, right next to nerve endings, for months after a lesion has resolved.
They stick around.
They stick around. Even during clinical quiescence, when there are no visible symptoms, these CD8+ T cells are actively making both antiviral and cytotoxic proteins, indicating ongoing immune surveillance.
Wow, constant vigilance.
It seems so. These resident memory CD8+ T cells seem to function as vigilant first responders, capable of controlling viral reactivation right at the site where the virus is released from the nerves into the dermis. This incredibly rapid, almost on and off interplay between the virus trying to reactivate and the host immune system helps explain the variability we see in clinical disease severity between episodes in any single individual.
Why some flare-ups are worse than others.
Exactly.
But
Consider this. Differences of merely 30 to 60 minutes in the host immune responses can result in 100 to 1,000-fold differences in viral levels at the surface.
just an hour makes that much difference.
It can. This subtle difference in the speed and effectiveness of the immune response can determine whether an episode of disease is contained subclinically without any visible symptoms or results in a full-blown clinical outbreak.
Fascinating.
There's a very strong association between the magnitude of the CD8+ T lymphocyte response and the clearance of virus from genital lesions. Therefore, the location, effectiveness, and longevity of these CD8+ T lymphocytes, along with other crucial immune influencers such as natural killer cells or CD4+ T cell responses, are likely critical factors in the expression of clinical disease and, importantly, the likelihood of transmission over time. 3. The global picture. Epidemiology, clinical spectrum, and vulnerable populations.
That dynamic dance between the virus and our immune system is truly a microdrama playing out in our bodies. Given how complex and pervasive the virus is in its biology, it's no surprise it's spread globally. What does the epidemiology tell us about its reach, and what are the big picture trends?
Indeed, it's a truly global challenge. Seroepidemiologic studies have documented HSV infections virtually worldwide. Over the past 15 years or so, a striking piece of data has emerged. The prevalence of HSV2 is actually higher in the developing world compared to developed nations.
Really? Why might that be?
The reasons are likely complex, involving socioeconomic factors, access to healthcare, maybe co-infections. For instance, in sub-Saharan Africa, HSV2 seroprevalence among pregnant women can approach an astonishing 60%.
60%.
Yes, with annual acquisition rates among teenage girls being very high. In contrast, in developed countries, the rate of HSV2 acquisition and serologic prevalence tends to be higher among women than among men. Interestingly, in middle and low-income countries, the frequency of genital HSV1 infection is currently low, and most HSV2 acquisition in these regions is preceded by HSV1 acquisition. Now, let's look at the acquisition patterns of HSV1 versus HSV2 more broadly. HSV1 is acquired more frequently and typically earlier in life.
Like in childhood.
Often, yes. By the fifth decade of life, between 70% and 90% of adults globally have antibodies to HSV1. In populations of lower socioeconomic status, most people acquire HSV1 infection even earlier, often before the third decade of life.
So almost everyone gets HSV1 eventually.
A very large proportion, yes. Antibodies to HSV2, however, are not routinely detected until puberty, and their prevalence rates directly correlate with past sexual activity, varying greatly among different population groups.
Okay.
In the U.S., serosurveys indicate that 15% to 20% of the overall population has antibodies to HSV2. In typical obstetric and family planning clinics, maybe 15% to 30% of women are HSV2 seropositive. Though it's striking that only about 10% of those who are seropositive for HSV2 actually report a history of genital lesions.
Most don't know they have it.
Or don't recognize the symptoms exactly. Among heterosexual adults attending sexually transmitted disease clinics, the figure can be as high as 50%. And this widespread prevalence is also mirrored in places like Central and South America and Africa, where HSV2 seroprevalence can range from 40% to 70% in sexually experienced populations, with rates typically 5% to 10% higher among women. Now, one of the most alarming, yet often unrecognized insights into HSV's global impact is its profound, almost sinister synergy with HIV-1.
Ah, the HIV link. I've heard about this.
Yes. Multiple studies consistently show that both incident meaning new and, more importantly, prevalent HSV2 infection significantly enhances the acquisition rate of HIV-1.
How much does it increase the risk?
On a population basis, HSV2 infection is associated with a 2 to 4-fold increase in HIV-1 acquisition. This has been amply demonstrated in heterosexual men and women in both developed and developing regions. Epidemiologically, areas of the world with a high HSV2 prevalence tend to have a higher population-based incidence of HIV-1. What's particularly critical to grasp here is how HSV2 facilitates the spread of HIV. Prevalent HSV2 appears to increase the risk of HIV infection by a substantial 7 to 9-fold on a per-coital basis.
7 to 9 times higher risk per sexual encounter. That's huge.
It's a staggering increase in risk, yes.
Right.
Mathematical models have even suggested that approximately 33% to 50% of HIV-1 infections globally may be attributable to HSV2, both in men who have sex with men and in heterosexual women, particularly in sub-Saharan Africa. Adding to this, HSV2 is more frequently reactivated in and transmitted by persons co-infected with HIV-1 compared to those who are not. It's a vicious cycle.
Makes sense.
This cyclical relationship contributes to why most areas of the world with a high HIV-1 prevalence also exhibit a high HSV2 prevalence. The mechanism is quite clear. The shedding of HIV-1 variants from herpetic lesions in the genital region directly facilitates the spread of HIV through sexual contact.
So the sores themselves are infectious for HIV too?
Potentially, yes. Furthermore, HSV2 reactivation causes a localized, persistent inflammatory response in the submucosa of the genital skin. This response consists of high concentrations of CCR5-enriched CD4 plus T cells.
The main target cells for HIV.
The primary targets, exactly, as well as inflammatory dendritic cells. These cells can readily support HIV infection and replication, thereby increasing the risk of HIV acquisition among persons with genital herpes. And here's a sobering point for us as professionals. Current antiviral therapy for HSV.
Like acyclovir.
Yes. While highly effective against HSV replication, unfortunately does not reduce the subclinical post-reactivation inflammation.
Really? Why not?
Well, it's likely because current antiviral agents can't fully prevent the release of small amounts of HSV antigen into the genital mucosa, which is enough to trigger that inflammatory response, even if they stop full lesion formation.
A crucial limitation.
It is. And this leads us to a major public health challenge: the asymptomatic reservoir and shedding dynamics. Several studies suggest that many cases of asymptomatic genital HSV2 infection are simply unrecognized by the individuals themselves.
Maybe the symptoms are mild, or in a place they can't see.
Exactly. It might be because the symptoms are so mild they go unnoticed, or the lesions are confined to anatomic regions of the genital tract that aren't easily visualized, such as the cervix or urethra. Critically, these asymptomatic seropositive persons shed virus on mucosal surfaces almost as frequently as do those with symptomatic disease.
Just as often. Wow.
Almost as often. This large reservoir of unidentified carriers of HSV2, coupled with the frequent asymptomatic reactivation of the virus from the genital tract, has consistently fostered the continued widespread global dissemination of genital herpes. HSV infections occur year-round, no seasonal variation. Transmission can happen through direct contact with persons who have active ulcerative lesions, but just as importantly, it also occurs from persons who show no clinical manifestations of infection, but are silently shedding HSV from mucocutaneous surfaces.
Silent spreaders.
Yes. Reactivation of HSV on genital skin and mucosal surfaces is remarkably common. In fact, recent studies indicate that most HSV1 and HSV2 shedding episodes are very brief, often lasting only 2 to 6 hours.
That incredibly short window again.
Exactly. This brevity means that detecting shedding accurately requires frequent sampling, often multiple times a day, to truly capture it. For HSV2, shedding occurs on about 10% to 30% of days.
Up to nearly a third of the time.
On any given day, yes. And for HSV1 in oral secretions, the figures are similar. Shedding rates are highest during the initial years after acquisition, with viral shedding occurring on as many as 30% to 50% of days during that period.
Half the time, potentially, early on.
Potentially. In immunosuppressed patients, the frequency is even higher, ranging from 20% to 80% of days. These high rates of mucocutaneous reactivation underscore why exposure to HSV from sexual or other close contact, like kissing or sharing utensils, is so common, and they help explain the continuing spread and high seroprevalence of HSV infections worldwide.
Right.
And while daily antiviral chemotherapy for HSV2 infection can significantly reduce shedding rates, it does not eliminate shedding entirely, as measured by PCR or culture.
Still some risk even on medication.
There is still some risk, yes. Among HIV-positive patients, factors influencing reactivation include a low CD4 plus T cell count and a high HIV-1 load, indicating weakened immune system makes the virus more active.
So, given how frequently the virus reactivates, even silently, what does that mean for the actual clinical picture? How does this virus manifest itself across the body, and are there certain populations particularly vulnerable?
Great question. The clinical manifestations of HSV are incredibly diverse. It really depends on the anatomic site involved, the age and immune status of the host, and critically, the antigenic type of the virus, whether it's HSV1 or HSV2.
So many variables.
So many variables. HSV has been isolated from nearly all visceral and mucocutaneous sites, highlighting its impressive versatility. Primary HSV infections.
The very first time you get it.
Right. The very first infections with either HSV1 or HSV2, where the host lacks pre-existing antibodies, are frequently accompanied by systemic signs and symptoms like fever or body aches.
Feeling generally unwell.
Exactly. Compared to recurrent episodes, these primary infections are typically longer in duration and often involve both mucosal and extramucosal sites. The incubation period for HSV can range from 1 to 26 days, with a median of 6 to 8 days. Now, while both HSV1 and HSV2 can cause both genital and oral facial infections, and clinically, the infections caused by the two subtypes are indistinguishable by appearance alone.
You can't tell just by looking.
You can't tell just by looking. But the frequency of reactivation is significantly influenced by both the anatomic site and the virus type. For instance, genital HSV2 infection is twice as likely to reactivate and recurs 8 to 10 times more frequently than genital HSV1 infection.
Much more frequent recurrences with genital HSV2.
Much more frequent. Conversely, oral labial HSV1 infection cold sores recurs more frequently than oral labial HSV2 infection. And as we just discussed, asymptomatic shedding rates follow precisely the same pattern. Let's quickly touch on some specific clinical syndromes. For oral facial infections, gingivostomatitis and pharyngitis are the most common clinical manifestations of a first episode HSV1 infection, especially among children and young adults.
Like really bad sore throat and mouth sores.
Yes, often presenting with systemic symptoms like fever, malaise, and difficulty eating, with lesions lasting days to weeks. The classic recurrent herpes labialis, or cold sores, is the most common manifestation of reactivated HSV1. This stems from the trigeminal ganglia and can lead to asymptomatic virus excretion in saliva or visible lip lesions. Surgical procedures, even dental extractions, are known triggers. For immunosuppressed patients, HSV infections can be particularly severe, extensive, and often difficult to manage. They can extend deep into mucosal and cutaneous layers.
Leading to bigger problems.
Yes, leading to friability, necrosis, severe pain, and significant difficulty with basic functions like eating. These lesions frequently occur alongside other infections, such as candida. Prophylactic systemic antiviral agents are absolutely crucial for these patients due to the exceptionally high frequency of HSV reactivation, 50% to 90%, during periods of severe immune suppression, like after transplantation or during chemotherapy.
So prevention is key there.
Prevention is absolutely key. HSV infections are also associated with other conditions, highlighting its broader impact on the body. Eczema herpeticum is a severe oral facial HSV infection seen in patients with atopic eczema. It can rapidly spread and in rare cases even disseminate to internal organs.
Dangerous in those patients.
Very dangerous. Erythema multiforme, a hypersensitivity skin reaction, is linked to HSV, with HSV being the precipitating event in about 75% of cutaneous cases. And HSV1 and VZV have been implicated in Bell's palsy, causing facial nerve paralysis, though antiviral benefit in trials has been mixed. Now, genital infections. A first episode of primary genital herpes is typically characterized by systemic symptoms like fever and body aches, alongside local pain, itching, and tender lymph nodes. Widely spaced bilateral lesions are characteristic.
On both sides?
Often on both sides initially, yes. In women, the cervix and urethra are involved in over 80% of first episodes. Recurrence rates are significantly higher for HSV2, around 90% within 12 months, compared to HSV1, about 55%.
Big difference.
Huge difference. HSV can also cause urethritis, endometritis, and even prostatitis. It's crucial to remember that about 15% of HSV2 acquisitions are associated with non-lesional syndromes, like aseptic meningitis or cervicitis, making diagnosis challenging.
So you might have HSV causing meningitis, but no sores.
That's possible, yes. It complicates the picture. Rectal and perianal infections can be symptomatic or asymptomatic proctitis, commonly associated with receptive rectal intercourse, but also from subclinical shedding from sacral latency. Symptoms include pain, discharge, and difficulty with bowel movements. These are especially common in immunosuppressed patients. Then there's herpetic whitlow.
On the finger.
Yes, an HSV infection of finger, typically occurring through inoculation, like a healthcare worker handling secretions or a child sucking their thumb. It presents with abrupt onset of swelling, redness, tenderness, and characteristic fascicular lesions. It often mimics bacterial infections, making prompt diagnosis crucial to avoid unnecessary surgery. Relatedly, herpes gladiatorum is seen in wrestlers, facilitated by skin trauma during matches, underscoring the importance of rapid diagnosis and treatment to contain outbreaks in close contact sports. Eye infections are a particularly serious concern. HSV infection of the eye is, remarkably, the most common cause of corneal blindness in the United States.
Corneal blindness.
Yes. HSV keratitis presents with pain, blurred vision, and characteristic dendritic lesions on the cornea. They look like little tree branches. It's vital to avoid topical glucocorticoids, as they can worsen the condition significantly.
Steroids make it worse.
They can, especially in early stages. Recurrences are common, and immunopathologic injury to deeper structures like stromal keratitis can occur, related to our own T cells trying to clear the virus but causing damage in the process. More severe manifestations include chorioretinitis and acute necrotizing retinitis, requiring emergent ophthalmology consultation. When HSV affects the central and peripheral nervous system, the consequences can be profound. HSV accounts for a significant 10% to 20% of all cases of sporadic viral encephalitis in the United States.
Inflammation of the brain.
Exactly. With HSV1 causing over 95% of cases, the clinical hallmark is acute fever and focal neurologic symptoms, particularly affecting the temporal lobe. Diagnosis relies heavily on PCR for HSV DNA in CSF.
From a spinal tap.
Yes, which has largely replaced brain biopsy. 4V acyclovir is critical and significantly reduces mortality, but neurologic sequelae, long-term brain damage, are common even with treatment. HSV DNA is also detected in 3% to 15% of cases of aseptic meningitis, usually with primary genital HSV. This is typically acute and self-limited. Notably, HSV is the most common cause of recurrent lymphocytic meningitis, known as Malariz meningitis.
Recurrent meningitis.
Yes, which can be managed with daily antiviral therapy for frequent recurrences. Autonomic radiculopathy and other PNS involvement, like transverse myelitis, Guillain-Barré syndrome, or Bell's palsy, are rare but serious neurological complications that can follow HSV infection, causing numbness, urinary retention, or paralysis. Finally, a fascinating and emerging area is the increasing experimental evidence suggesting an association between HSV1 and the development of sporadic Alzheimer's disease. The Alzheimer's link. This is controversial, right?
It is, and it's still very much in the research phase. HSV1 DNA has been detected in the brain tissue of Alzheimer's patients, and HSV1 antibodies are a significant risk factor for later onset of the disease. While models show HSV1 inducing neuronal death and amyloid plaque-like structures, it is crucial to state that currently no cogent data indicates antiviral therapy would benefit anyone with Alzheimer's disease.
So no reason to treat AD with acyclovir based on this?
Absolutely not based on current evidence. This is an active area of research, not a current treatment pathway. Moving to visceral infections, HSV infection of internal organs usually results from viremia and often leads to multiple organ involvement, but can be limited to just one. HSV esophagitis causes painful swallowing and is diagnosed by cytology, culture, or PCR of secretions. Systemic antiviral therapy is effective. HSV pneumonitis is uncommon, except in severely immunosuppressed patients, and carries a high mortality. HSV found in tracheal aspirates of ARDS patients is often localized tracheitis reactivation, not true pneumonia, though its overall impact is unclear. HSV hepatitis is rare in immunocompetent patients, but causes fever, elevated liver enzymes, and other systemic issues. Other rare complications include monoarticular arthritis and glomerulonephritis. Disseminated HSV.
Spreading everywhere.
Spreading everywhere. It's rare in immunocompetent people, but occurs in immunocompromised patients and can be life-threatening, especially when primary infection occurs in pregnancy. It's best detected by HSV DNA in plasma or blood. Now, let's turn our attention to neonatal HSV infections, which are a critical concern. Among all HSV-infected populations, neonates infants under 6 weeks of age have the highest frequency of visceral and/or central nervous system infection.
The most vulnerable group.
By far. Without therapy, the overall mortality is a devastating 65%. And tragically, fewer than 10% of neonates with CNS infection develop normally, highlighting the severe long-term neurological morbidity. This is a truly dire prognosis. While skin lesions are the most commonly recognized features of neonatal disease, it's crucial for professionals to understand that many infected infants do not develop lesions at all, or they develop them only well into the course of the disease, making early diagnosis incredibly challenging.
So you can't rely on seeing sores.
You absolutely cannot rely on seeing sores. Neonatal infection is typically acquired perinatally, meaning during birth, from contact with infected genital secretions during delivery. The breakdown is roughly 30% to 50% due to HSV1 and 50% to 70% due to HSV2. While less common, congenitally infected infants, meaning infection acquired in utero, have also been reported. The risk of an infant developing neonatal HSV infection is significantly higher, about 10 times higher, for an infant born to a mother who has recently acquired HSV compared to an infant born to a mother with recurrent HSV.
So a new infection in the mother near delivery is the highest risk.
By far the highest risk. Transmission can also occur from a mother's oral labial HSV1 infection, or even through nosocomial transmission within the hospital setting, though these are rarer.
Okay.
The treatment paradigm is clear and urgent. All neonates with presumed herpes should be treated immediately with intravenous acyclovir and then placed on maintenance oral antiviral therapy for the first 6 to 12 months of life.
Aggressive treatment.
Very aggressive and vital. Antiviral chemotherapy with high-dose intravenous acyclovir at 60 mg/kg per day has significantly reduced the mortality rate from neonatal herpes to approximately 15%.
That's a big improvement.
A huge improvement in survival. However, despite this improvement, rates of morbidity, especially among infants with HSV2 infection involving the central nervous system, remain very high. It underscores the ongoing challenge and the need for continued vigilance. Moving to HSV in pregnancy in the United States, about 21% of all pregnant women are seropositive for HSV2. That figure rises to 51% among non-Hispanic Black pregnant women.
Very common.
Very common. The risk of mother-to-child transmission of HSV in the perinatal period is highest when the infection is acquired near the time of labor, particularly in women who were previously HSV seronegative.
That high-risk scenario again?
Exactly. The clinical manifestations of recurrent genital herpes, including frequency, duration, pain, are similar in both pregnant and non-pregnant women. Interestingly, recurrences actually tend to increase in frequency over the course of pregnancy. However, when women are seropositive for HSV2 at the outset of pregnancy, there is no discernible effect on neonatal outcomes, like birth weight or gestational age. First episode infections acquired during pregnancy carry more severe consequences for both the mother and the infant. Maternal visceral dissemination during the third trimester can occasionally occur, as can premature birth or intrauterine growth retardation. The acquisition of primary disease in pregnancy carries a rare but serious risk of transplacental transmission.
Across the placenta.
Yes, across the placenta to the neonate, which can result in spontaneous abortion. For newly acquired genital HSV infection during pregnancy, most authorities recommend treatment with acyclovir or valacyclovir for 7 to 10 days.
To treat the mother.
Primarily for maternal benefit, yes. It's important to note that the impact of this intervention on preventing transmission to the infant is currently unknown. critically, no adverse fetal or newborn effects have been definitively attributed to acyclovir based on extensive data.
So acyclovir is considered safe in pregnancy?
Yes, it is considered safe for use at all stages of pregnancy and by breastfeeding women. The drug can be found in breast milk, but without reported harm to the infant. Given the high HSV2 prevalence rate in pregnancy and the relatively low incidence of neonatal disease, approximately 1 case per 6,000, 20,000 live births cesarean section, is not warranted for all women with recurrent genital disease.
Right, that would be a lot of unnecessary C-sections.
Exactly. Since interpartum transmission during delivery accounts for the majority of cases, abdominal delivery needs to be considered only for women who are actively shedding HSV at the time of delivery. Several studies have shown no correlation between the recurrence of viral shedding before delivery and viral shedding at term.
So testing beforehand doesn't predict shedding at delivery?
Not reliably, no. Therefore, weekly virologic monitoring, or amniocentesis, are not recommended as routine practices. They don't reliably predict shedding at delivery. The frequency of transmission from mother to infant is markedly higher among women who acquire HSV near term, ranging from 30% to 50%. Compare that to those in whom HSV2 infection is simply reactivated at delivery, which carries a risk of less than 1%.
Huge difference based on timing of maternal infection.
Massive difference. While maternal antibody to HSV2 is protective, antibody to HSV1 offers little or no protection against neonatal HSV2 infection. Interestingly, primary genital infection with HSV1 leads to a particularly high risk of transmission during pregnancy and accounts for an increasing proportion of neonatal HSV cases.
So primary HSV1 is also a big risk factor?
Yes. Moreover, during reactivation, HSV1 appears to be more transmissible to the neonate than HSV2. Despite the low frequency of transmission of HSV when mothers have established genital herpes, only 2% of HSV2 seropositive women have HSV2 isolated from cervical secretions at delivery, and only 1% of infants exposed in this manner develop infection. It's important to note that 30% to 50% of infants with neonatal HSV are actually born to mothers with established genital herpes.
How does that work if the risk is so low per birth?
It suggests that even these low-level, often asymptomatic shedding events contribute significantly to the overall burden just because established herpes is so common. It makes identification and prevention a real challenge. The isolation of HSV by cervicovaginal swab at the time of delivery is the greatest risk factor for intrapartum HSV transmission, carrying an astonishing relative risk of 346. It's also known that culture-negative but PCR-positive cases of intrapartum transmission are well described, highlighting the superior sensitivity of PCR. Other risk factors for transmission include new acquisition of HSV, odds ratio 49, isolation of HSV1 versus HSV2, or R35, detection of HSV in cervical versus vulvar lesions, or R15, the use of fetal scalp electrodes during labor, or R3.5, and young maternal age. Conversely, cesarean delivery is highly protective with an odds ratio of 0.14.
C-section definitely helps if shedding is happening.
Yes. Physical examination alone is a poor predictor of the absence of shedding, and PCR far exceeds culture in terms of sensitivity and speed for detecting viral presence. Therefore, while PCR detection at the onset of labor is a promising area to reduce maternal fetal transmission, patients with recurrent genital herpes should be encouraged to come to the hospital early at the time of delivery for a careful examination of the external genitalia and cervix, as well as collection of a swab sample for viral isolation.
Check for lesions right at delivery time.
Exactly. If there is no evidence of active lesions, a vaginal delivery can proceed. However, the presence of active lesions on the cervix or external genitalia is a clear indication for cesarean delivery. For infant care post-delivery, if first episode exposure has occurred, for example, if the mother is seronegative, or if the mother is HSV1 seropositive and the isolated delivery is found to be HSV2, many authorities would initiate antiviral therapy for the infant with intravenous acyclovir immediately.
Treat the baby right away in high-risk situations.
Yes. At a minimum, samples for viral cultures and PCR should be obtained from the infant's throat, nasopharynx, eyes, and rectum immediately after birth and at 5 to 10 day intervals to monitor for infection. Any signs of lethargy, skin lesions, or fever in the infant should trigger an immediate evaluation. And critically, all infants from whom HSV is isolated 24 hours after delivery should be treated with intravenous acyclovir at the recommended doses, regardless of clinical symptoms. 4. Diagnosis and treatment. Mastering management.
That brings us to a crucial point for professionals. If you suspect HSV, especially with all these nuances between clinical presentation and that silent shedding we talked about, how do you confirm it accurately and reliably?
That's a critical question, and it really separates general awareness from professional mastery. Both clinical and laboratory criteria are used for diagnosing HSV infections. A clinical diagnosis can be made with reasonable accuracy when the characteristic multiple vesicular lesions on an erythematous base are present.
The classic look.
The classic look, yes, that blister on a red background. However, herpetic ulcerations can often resemble skin ulcerations caused by other etiologies, and mucosal HSV infections can present as urethritis or pharyngitis without any visible cutaneous lesions.
So looks can be deceiving.
They absolutely can be. This is why laboratory studies to confirm the diagnosis and to guide therapy are strongly recommended. While older cytologic methods, such as staining scrapings from the base of the lesions like the Zank preparation, or pap smears to detect giant cells or intranuclear inclusions, are well-described procedures.
Things you read about in textbooks.
Exactly. But few clinicians are actually skilled in these techniques anymore. Furthermore, their sensitivity is quite low, less than 30% for mucosal swabs, and they cannot reliably differentiate between HSV and varicella-zoster virus, or VZV, which can cause similar-looking lesions. Therefore, the professional standard now is that HSV infection is best confirmed in the laboratory by directly detecting the virus, viral antigen, or, most commonly and sensitively, viral DNA in scrapings from lesions.
PCR testing.
HSV DNA detection by PCR, or polymerase chain reaction, yes. It is the most sensitive laboratory technique for detecting mucosal or visceral HSV infections, and it is unequivocally the recommended test for laboratory confirmation of a diagnosis due to its unparalleled speed and sensitivity.
Okay. Is culture still used?
For situations where you might need to test for drug resistance, cell culture remains useful. HSV causes a discernible cytopathic effect in a variety of cell culture systems, meaning it physically changes the cells in a way we can see under a microscope, usually within 48 to 96 hours. Spin-amplified culture with subsequent staining for HSV antigen can even shorten this time to less than 24 hours.
Got it.
It's important to remember that the sensitivity of all detection methods depends on several factors. The stage of the lesions. Vesicular is better than ulcerative.
Get the sample early.
Get it early. Whether the patient has a first or a recurrent episode, first episodes usually have more virus, and the patient's immune status. Immunosuppressed patients tend to shed more virus. Laboratory confirmation is also invaluable because it permits subtyping of the virus.
Telling HSV1 from HSV2?
Right, providing information that can be useful epidemiologically and help predict the frequency of reactivation after first episode oral, labial, or genital HSV infection. Knowing if it's HSV1 or HSV2 from the start can help set expectations for the patient. Beyond acute diagnosis from a lesion, serologic assays, the blood tests, play a vital role, especially in understanding past exposure. Both type-specific and type-common antibodies to HSV develop within the first several weeks after infection and persist indefinitely.
Okay.
Traditional serologic assays that use whole virus antigen preparations are useful for differentiating uninfected individuals from those with past HSV1 or HSV2 infection. However, they do not reliably distinguish between the two viral subtypes because of that significant antigenic cross-reactivity we discussed. Your body just sees herpes broadly.
So the older tests weren't great for subtyping.
Not reliable for subtyping, no. Crucially, for an accurate professional assessment, type-specific glycoprotein G assays, specifically G1 for HSV1 and G2 for HSV2, are commercially available and can reliably distinguish between human antibody responses to HSV1 and HSV2.
Glycoprotein G, the key difference.
Exactly. Point-of-care assays, which provide results from capillary blood or serum during a clinic visit, are also available, as is a Western blot assay that can detect several HSV type-specific proteins. The presence of type-specific HSV2 antibody reliably implies past HSV2 infection, meaning a latent infection and very likely ongoing subclinical reactivation.
Okay.
While acute and convalescent phase serum samples can be useful in diagnosing primary infection, there are caveats. Acute phase samples may lack antibodies, and what appears to be a first episode can occur in individuals who are already seropositive for the other type of HSV. Therefore, type-specific protein assays should be the go-to for identifying asymptomatic carriers of HSV1 or HSV2. It's also important to note that no reliable IgM method for defining acute HSV infection is currently available for routine use. For empowering the listener you with this knowledge, it's worth noting that studies have shown that persons with previously unrecognized HSV2 infection can be taught to identify their own subtle symptomatic reactivations.
People can learn to spot their own symptoms better.
They can, and it's a public health imperative. Individuals who are seropositive for HSV2 should be informed about the high frequency of subclinical reactivation on mucosal surfaces that are not visible to the eye, like the cervix, urethra, or perianal skin, or in microscopic ulcerations that may not be clinically symptomatic. Transmission of infection during these subclinical episodes is well established.
Right.
Therefore, HSV-2 seropositive persons should be educated about the high likelihood of subclinical shedding and the important role that condoms, both male and female, can play in reducing transmission. Furthermore, a key clinical trial result: antiviral therapy with valacyclovir, specifically 500 milligrams once daily, has been clinically shown to reduce the transmission of HSV-2 between sexual partners.
So daily meds can reduce spreading it.
Yes, that's actual information based on trial data.
Okay, so we've covered how to accurately identify it with these increasingly precise tools. Now, for the real fight, how do we combat this master of persistence? What's in our antiviral arsenal, and how do these drugs actually work?
The good news is that many aspects of mucocutaneous and visceral HSV infections are very amenable to antiviral chemotherapy. For mucocutaneous infections, acyclovir and its close relatives, acyclovir and valacyclovir, have been the mainstays of therapy for decades.
Acyclovirs?
The cyclovirs, yes. For HSD-I infections, several antiviral agents are available for topical use, including idoxuridine, trifluorothymidine, topical vidarabine, and cidofovir. But for the most severe manifestations like HSV encephalitis and neonatal herpes, intravenous acyclovir is the treatment of choice, highlighting its critical, life-saving role in reducing death and severe morbidity. The mechanism of action for all licensed antiviral agents against HSV is quite elegant, actually. It's a classic example of targeted therapy. They all specifically inhibit viral DNA polymerase.
The enzyme that copies the virus's DNA.
Exactly. Drugs like acyclovir, ganciclovir, famciclovir, and valacyclovir are all designed to be selectively phosphorylated to their monophosphate form only within virus-infected cells.
So they only get activated inside infected cells.
Precisely. This selectivity is thanks to the HSV enzyme thymidine kinase, or TK, which is primarily found in infected cells. This makes the drug essentially invisible to uninfected cells until the virus activates it.
Very clever drug design.
It is. Once monophosphorylated, our own cellular enzymes then convert this monophosphate form of the drug into its triphosphate form. This triphosphate is then incorporated into the viral DNA chain, effectively acting as a molecular stop sign that halts viral DNA replication. This is how these drugs stop the virus from making copies of itself without harming our healthy cells significantly. Let's quickly review the main drug profiles and their critical clinical applications. Acyclovir is the agent most frequently used for HSV infections and is available in intravenous, oral, and topical formulations.
The original.
The original, yes. Valacyclovir is a valacylase of acyclovir, which offers significantly greater bioavailability when taken orally.
Meaning more gets absorbed.
Right, more of the drug gets into your bloodstream, allowing for less frequent administration. Famciclovir is the oral formulation of penciclovir, and it's also clinically effective for a variety of HSV-1 and HSV-2 infections. Ganciclovir, while active against both HSV-1 and HSV-2, is generally more toxic than the others, and is usually not recommended for typical HSV infections unless dealing with very specific resistant strains.
Okay.
All three recommended compounds, acyclovir, valacyclovir, and famciclovir, have proven highly effective in shortening the duration of symptoms and lesions of mucocutaneous HSV infections in both immunocompromised and immunocompetent patients. These antiviral agents are also critical for prophylaxis in immunocompromised patients.
Preventing flare-ups.
Exactly. Intravenous and oral formulations are routinely used to prevent HSV reactivation in seropositive immunocompromised patients during periods like induction chemotherapy or immediately after bone marrow or solid organ transplantation, where their immune systems are severely suppressed. This preventive use significantly reduces the burden of HSV disease in this vulnerable population. Chronic daily suppressive therapy is also a cornerstone of management, significantly reducing the frequency of reactivation disease for patients who experience frequent genital or oral labial herpes. In terms of public health impact, it's notable that only valacyclovir has been subjected to large-scale clinical trials that specifically demonstrated its ability to reduce the transmission of HSV-2 infection between sexual partners.
Only valacyclovir has that specific data for transmission reduction.
Correct, based on published large trials. For HSV encephalitis, intravenous acyclovir at a dosage of 30 milligigaograms per day, given in 3 divided doses, is highly effective in reducing rates of death and morbidity. The early initiation of therapy is the critical factor in determining the neurological outcome and survival.
Gotta start it fast.
Absolutely critical to start it fast. A major side effect associated with intravenous acyclovir is transient renal insufficiency, usually due to the crystallization of the compound in the renal parenchyma. This can largely be avoided if the medication is infused slowly over 1 hour and the patient is well hydrated. It's important for professionals to note the dosage difference, because acyclovir CSF levels average only 30% to 50% of plasma levels. The dosage for CNS infection, 30 kilograms per day, is much higher than for mucocutaneous infection, typically 15 kilograms per day, and for neonatal HSV infection, a whopping 60 milligrams per day in 3 divided doses, due to the severity and critical nature of these infections.
Higher doses needed for the brain and babies.
Exactly. Despite their impressive efficacy, these antiviral drugs have important limitations, and this is a key takeaway. They do not eradicate latent infection.
They don't cure it.
They do not cure it. Nor do they affect the risk, frequency, or severity of subclinical or clinical recurrence after the drug is discontinued. The virus remains in its hidden state, ready to reactivate.
Okay.
For immunocompetent patients, shorter courses of therapy are increasingly being used for recurrent mucocutaneous infection. One-day courses of famciclovir and valacyclovir have been shown to be clinically effective, more convenient, and generally less costly than longer regimens, making them a preferred option for this population due to improved adherence.
Shorter treatment for outbreaks is good.
Very convenient, yes. The recognition of the high frequency of subclinical reactivation provides a well-accepted rationale for the use of chronic daily suppressive therapy, especially for persons with frequent clinical reactivations, such as those with recently acquired genital HSV infection. Immunosuppressed individuals, including those with HIV infection, also benefit significantly from daily antiviral therapy.
Daily meds for frequent recurrences or immune issues.
Correct. Daily acyclovir and valacyclovir can substantially reduce the frequency of both clinical and subclinical HSV reactivations among HIV-positive persons. Common regimens include acyclovir, 400 to 800 milligrams twice daily, famciclovir, 500 milligrams twice daily, and valacyclovir, 500 milligrams twice daily. It's worth noting that valacyclovir at a very high dose of 4 grams per day was associated with TTP, a serious blood disorder, in one study of HIV-infected individuals, so dosage must be carefully managed in that specific population.
Okay, caution needed there.
Yes. Interestingly, daily acyclovir therapy has also been associated with a modest reduction in the titer of HIV RNA in plasma, about a 0.5 log 10 reduction and a 0.33 log 10 reduction in the genital mucosa.
So it has a slight anti-HIV effect too.
A modest effect, yes. It hints at a complex interaction that we're still trying to fully unravel. The impact on public health is significant when we consider the reduced HSV transmission to sexual partners. As we mentioned, once daily valacyclovir at 500 milligrams has been shown in clinical trials to reduce the transmission of HSV2 between sexual partners. Transmission rates are generally higher from males to females and among persons with frequent HSV2 reactivation. Serologic screening can be a valuable tool to identify at-risk couples who might benefit from this intervention.
So testing couples can help guide prevention.
It can. While both valacyclovir and famciclovir are effective for suppression, valacyclovir appears to be more effective at reducing subclinical shedding compared to famciclovir based on available data. However, a growing challenge in HSV management is acyclovir resistance. Clinically relevant acyclovir-resistant HSV strains do occur, and this is something professionals need to be acutely aware of.
The drugs stop working.
Essentially, yes. Most of these resistant strains have an altered substrate specificity for phosphorylating acyclovir, meaning the viral thymidine kinase enzyme, the TK enzyme, is changed and doesn't activate the drug as effectively.
So the first step in activation failed.
Exactly. This often leads to cross-resistance to famciclovir and valacyclovir as well, because they rely on the same activation pathway. Occasionally, an isolate with altered TK specificity may arise that is sensitive to famciclovir but not to acyclovir. In some patients infected with TK-deficient virus, higher doses of acyclovir might be associated with clearing of lesions, but in others, clinical disease progresses despite high-dose therapy, a clear sign of resistance.
Where do you see resistance most often?
The majority of clinically significant acyclovir resistance has been observed in immunocompromised patients. And HSV2 isolates generally appear to be more resistant than HSV1 strains. The overall frequency of acyclovir resistance isn't well characterized or consistently monitored globally, but the lack of appreciable change over the past 40 years likely reflects the reduced transmission of these TK-deficient mutants.
Why are they less fit?
They seem to be less capable of spreading efficiently in the general population, likely due to altered neurovirulence. Suspicion of resistance should arise whenever HSV lesions persist, despite adequate dosages and documented blood levels of acyclovir. That's a clear red flag for clinicians. The clinical management of acyclovir resistance is quite challenging. therapy with the antiviral drug foscarnet, administered intravenously.
A different kind of drug.
Yes, a different mechanism. Given AVI at 40 to 80 million kilograms every 8 hours until clinical resolution, it's currently the only clinically demonstrated approach for resistant HSV. However, because of its potential for toxicity, particularly to the kidneys, and its high cost, foscarnet is usually reserved for patients with extensive mucocutaneous infections that are truly refractory to standard therapy.
So it's a second-line heavy hitter.
Exactly. Cidofovir, a nucleotide analog, is active against most TK-deficient strains of HSV, and its ointment formulation has been shown to speed the healing of acyclovir-resistant lesions, though well-controlled systemic trials are lacking. Topical imiquimod, an immune response modifier, can also be considered in some cases. It's important to remember that true TK-negative variants of HSV appear to have a reduced capacity to spread due to altered neurovirulence, which helps explain the relatively infrequent presence in immunocompetent populations, even with the increasing use of antiviral drugs.
Okay, any new drugs coming?
Looking to the future, a new class of drugs that inhibit HSV-specific helicosampomas activity, such as white liver, is currently under clinical investigation. These might offer a better toxicity profile for the treatment of acyclovir-resistant HSV infection. That would be a significant breakthrough. On the topic of acyclovir and pregnancy, there's very reassuring news for healthcare providers and expectant mothers. No adverse effects to the fetus or newborn have been reliably attributed to acyclovir.
Good news there.
Very good news. It can be used safely at all stages of pregnancy and by women who are breastfeeding, as the drug can be found in breast milk, but without reported harm. Suppressive acyclovir treatment in late pregnancy, for example, acyclovir 400 milligrams orally 3 times daily, or valacyclovir 500 milligrams orally twice daily from around 34 weeks until delivery, has been shown to reduce the frequency of cesarean delivery among women with recurrent genital herpes.
By preventing outbreaks at delivery time.
Precisely. However, it's important to clarify that such treatment may not independently protect against transmission to neonates. Its primary benefit is reducing the need for C-section by suppressing visible lesions at term. Finally, there are also global efficacy disparities to consider, which is a significant point for understanding HSV's worldwide impact. Initial studies of acyclovir-like drugs were performed primarily in the developed world.
Right.
While acyclovir, valacyclovir, and famciclovir are undeniably effective in the developing world, their clinical and virologic benefits, particularly in reducing the frequency of genital lesions among patients in African populations, seem to be reduced compared to what is observed in European and U.S. populations.
Less effective in some populations. Why?
The mechanism underlying this phenomenon is currently uncertain. It's an active area of research, could be host genetics, viral strains, co-infections. We don't fully know. Curiously, while daily acyclovir modestly reduces HIV RNA, the anti-HIV drug tenofovir has been shown to reduce HSV2 acquisition among women in Africa, but it has no demonstrable clinical benefit or antiviral effects among persons with established HSV2 infection in studies conducted in the United States.
That's weird. Tenofovir prevents HSV2 there, but doesn't treat it here.
The reasons for these disparate results are also unclear. It highlights the complexities of viral interactions in different populations and the need for region-specific research. When it comes to public health and future needs, efforts to control HSV disease on a population basis through widespread suppressive antiviral therapy and/or broad educational programs have been limited in their overall impact so far.
We haven't really gotten a handle on it globally.
Not as much as we'd like. Barrier forms of contraception, especially condoms, have been shown to decrease the likelihood of HSV infection transmission, particularly during periods of asymptomatic viral excretion. Even when lesions are present, HSV infection may be transmitted by skin-to-skin contact despite condom use. But available data consistently suggests that consistent condom use is an effective means of reducing the risk of genital HSV2 transmission.
Condoms help, especially with silent shedding.
Yes. Chronic daily antiviral therapy with valacyclovir can also be partially effective in reducing the acquisition of HSV2, particularly among susceptible women. Since there are no comparative efficacy studies directly comparing valacyclovir use versus condom use, most authorities suggest utilizing both approaches for optimal prevention. Perhaps the most urgent public health need in this entire arena is for a vaccine to prevent the acquisition of HSV infection.
The ultimate goal.
The ultimate goal, especially given the significant role that HSV2 plays in enhancing the acquisition and transmission of HIV-1, as we discussed earlier. A substantial portion of neonatal HSV cases could be prevented by reducing the acquisition of HSV by women in the third trimester of pregnancy.
Avoiding that high-risk late pregnancy infection.
Exactly. Neonatal HSV infection can result either from the acquisition of maternal infection near term or the reactivation of infection at delivery in an already infected mother. Therefore, women without known genital herpes should be counseled to abstain from vaginal intercourse during the third trimester with partners known to have or suspected of having genital herpes.
Practical advice.
Yes. While some authorities have recommended antiviral therapy with acyclovir or valacyclovir for HSV2 infected women in late pregnancy as a means of reducing HSV2 reactivation at term, data are not available to fully support the efficacy of this approach in definitively preventing neonatal transmission.
Yeah.
Moreover, the high treatment to prevention ratio.
You have to treat many women to prevent one case.
Exactly. It makes this a difficult, if not dubious, public health strategy solely for prevention, even though it can reduce the frequency of HSV-associated cesarean delivery.
What a journey. We've truly taken a deep dive into herpes simplex virus infections today. From the incredible molecular details of its life cycle and its clever, almost cunning ways of evading our immune system, to its pervasive global impact and the complex strategies involved in diagnosis and treatment, it's clear this virus is a master of persistence.
It really is. If we connect this to the bigger picture, the persistent challenges of HSV, particularly its remarkable ability to establish latency and shed subclinically, underscore why it remains such a prevalent and complex issue despite decades of research and highly effective antiviral medications.
Yeah.
It's a testament to the virus's evolutionary success, really,
Yeah.
and a constant reminder of how adaptable pathogens can be, always finding a way to survive.
This raises an important question for us and for you, our dedicated learner, to continue your mastery. Considering the intricacies of latency, the ongoing global burden, and those fascinating but also frustrating immune evasion tactics, what scientific or public health breakthroughs do you think are truly needed for us to achieve a new level of control? Perhaps even, dare I say, eradication of HSV infections.
That's the billion-dollar question.
Right. Think about the potential of novel gene editing technologies to truly interrupt that viral hide-and-seek game, or perhaps broad-spectrum immune-modulating therapies that could prevent reactivation entirely. There's always more to learn and explore, and the story of HSV is clearly far from over.
Indeed. Understanding it deeply, as we've aimed to do today, is truly the first step in contributing to its future and perhaps its ultimate defeat.